anatomists and physiologists. This video is a continuation of our digestive organs part 2 video. So let's recall what we ate and swallowed food. When we ate and swallowed that food on down the line, it traveled down the esophagus, passed into the stomach with a bunch of events happening there, and now that leads us into the small intestine, which is all of this stuff. And the small intestine is pretty long, so there's some regions within the small intestine to kind of differentiate between all of the length. So here are those regions that we're talking about. The first part, and I'll highlight as we go, the first part of the small intestine is called the duodenum. The duodenum of the small intestine is actually the shortest part. It's just this purple curved bit. And then the pancreas is kind of like nested in that little curve. So from the pyloric sphincter, then we lead into the duodenum. After the duodenum is this whole blue section that's called the jejunum. From the jejunum, we lead into the ileum, which is the pink part that's last. And then that ileum leads us into large intestine. The small intestine has a lot of special structures that make it a really great absorber of whatever we've digested. The thing with the small intestine is that it's always about increasing surface area so we can be good at absorption. Let me show you what I mean. So this is the tube of the small intestine. So here's the small intestine. And remember that it's still part of the GI tract, so there's still those major 4 layers. There's still serosa, there's still muscularis externa, submucosa, mucosa. But there's some extra bits here to discuss. The first thing to note here is that the small intestine, like I said, is a great absorber of nutrients. So having more ways to absorb better is the name of the game. And there's a few major ways this happens. So we're looking here, all of this little these rings that are made inside are circular folds. That's what these guys are. So circular folds, it's a new word, I'm highlighting it. These guys slow the flow of the contents in the small intestine. Slow the flow. If things move slower through the small intestine, we have more time for absorption. And it makes sense because what happens here is that everything that's in the small intestine kind of circles around and around and around. And it's have you ever been up a spiral staircase? It takes a long time to do that compared to just a staircase that's a straight shot. The other stuff that we have to talk about is increasing that surface area. So inside of the intestine's surface area, if we were to measure it all out, it would be 200 square meters, which is 656 square feet. That's like a small apartment. It's not small in there. The next thing we have to discuss is this next new word called villi. Villi are these little finger-like projections. All of those little finger-like projections create little extensions, and all of those things increase surface area. Villi are most abundant in the duodenum because that's where most absorption happens. The next things are microvilli, which are just like their name says, they're small villi. But I'll show you a zoomed in picture of that so we can see what we're talking about. Okay, so what I was talking about are villi. So villi are all of these guys. That's a villus, that's a villus, that's a villus. So singular, villus, multiple villi. In each villus, if we were to zoom in, is a bunch of cells. So here's just one of those cells. And these little furry little projections on there, that's the microvilli. Microvilli are just extensions of the plasma membrane that go up and down and up and down and up and down and up and down in order to increase surface area. And on microscope, microvilli look kind of fuzzy, so they call it the brush border. So brush border, that's another important term. The cells here also make enzymes. So there's also enzymes here that aid in digestion for carbs, and proteins. And they're called brush border enzymes. So now that we've zoomed so far into the small intestine, it's a good time to discuss what kinds of cells and other stuff that are hanging out here. Okay, so we have the microvilli, we have the brush border, but we also have these cells called enterocytes. Entero, remember, refers to gut. Cytes refers to cell. So they're gut cells, intestinal cells. They're most abundant, and it's because they're absorbing cells. They take the nutrients. The bottom of the villi are called intestinal crypts. So if we go to the bottom here, those are called intestinal crypts. That word is right here, intestinal crypt. So we go all the way to the bottom there. So if enterocytes are found there, they make intestinal juice that's kind of like a mucusy, and it helps with absorbing stuff. Also in here, we have goblet cells. Goblet cells make mucus. And that's really important for doing all of the things that we need to do and also being just protective because it's a harsh environment there. We have more enteroendocrine cells. Entero meaning gut, endocrine meaning hormones and stuff. And that helps regulate the activities of the GI tract. Regulate GI activity. We also have Paneth cells. Paneth cells live in the crypts, and they make antimicrobial agents. Antimicrobial agents. Like defensins, like lysozyme, and that helps protect from pathogens, and they can destroy certain bacteria. Note that I said some bacteria, but not all. Some bacteria are actually really good, and we'll talk about them later. Lastly, we have stem cells in here. And the stem cells, those are good because they make sure that there's a healthy supply of these cell types. Because what happens is after a while, these cell types get sloughed off. They're made down here, like way in the bottoms, and then they slowly move up and then they get sloughed off. So these cells are replaced like every 3 to 5 days. But we also have those lymph functions here. We have that malt tissue, mucosa-associated lymphoid tissue. That's malt. And like our Peyer's patches, right? So there's lymphoid function there to help protect against pathogens, but also part of the lymphatic system are lacteals. Lacteals are specialized lymph vessels for picking up large lipid molecules to be broken down into smaller and smaller bits by other organs. But we'll chat about that. So all of that anatomy and histology leads us to what does the small intestine actually do? What digestive processes go on here? We move stuff through and toward the end, but mostly what happens instead of propulsion in the small intestine, it's not so much propulsion, it's really more segmentation. I put a line through propulsion just because it's really slow. It doesn't really occur quickly. And that's good because we want enough time to absorb all of the things, right? Um, but that segmentation is really good because we need to do lots of mixing. We have to think about this, right? Because the chyme coming from the stomach needs to mix with the contents of the things that are dumped into the small intestine. Because we also have small intestine input from the bladder, or I mean the gallbladder, which is bile, and from the pancreas, and then also with its own intestinal juices. And so that peristalsis function is a lot more mild compared to what we see in the stomach. Along, also different from the stomach is the digestive juices. Gastric juice is acidic, hydrochloric acid and stuff. Intestinal juices are very basic. So there's a lot of adjustment that needs to be made in order for those pHs to balance out. So again, this is mixing and segmentation. A lot of chemical digestion also happens here. Remember that the stomach did some, but the small intestine kind of tries to like finish the job. There's lots of enzymes that aid in this, and a lot of those enzymes come from accessory organs. Most enzymes come from accessory organs, especially the liver and the pancreas. But there's also the brush border enzymes, which do the carbs and the proteins. Absorption happens a lot in the small intestine, especially in the duodenum. Duodenum. That's like the most. And that passage through the small intestine usually takes several hours. So we hang out, we digest, we absorb, we have a good time. Like that's the focus. So that's what the small intestine does. But how does that get regulated? The first layer is by regulating the entry of chyme into the small intestine. And that's important because chyme is usually what's called hypertonic, which just means that there's lots of solutes in there. So I'll write this down. Chyme is hypertonic, which means that there's lots of solutes in chyme. When there's a lot of solutes, that's going to draw water and it's going to get the water from our blood, which means that we're going to pull water from the blood and we're going to lose water from the blood by the process of osmosis. And that's going to decrease blood volume because all of that water is going to be shoved into the intestinal lumen. And we really don't want that. But also, chyme is very acidic. While the intestinal juices are very basic, so we need to regulate those pHs. And that takes time because we have to mix the chyme with the pancreatic juice and with the bile from the gallbladder. And there's a reflex. It's called the enterogastric reflex. Enterogastric reflex, which makes sure that the duodenum, the first portion of the small intestine, isn't overloaded with too much chyme at any one time. Motility is the other thing. Motility is the other thing. There's segmentation or mixing. That's the main thing that happens while we're actively digesting and absorbing, like after a meal. The stomach and the small intestine both have their own rhythms at which they do their contractions for peristalsis or segmentation. And that causes that movement for the small intestine to be really slow after a meal because it's good because we want that time to absorb. That's really important. So motility is more segmentation, less moving through peristalsis. Between meals, though, we do have peristalsis kicking up a little, And that's because we want things to move a little bit faster through the small intestine while we're not actively digesting and stuff. And that's because the small intestine, we want to kind of like sweep the bits of chyme that's left over, any bacteria, any dead cells. We want to put all that in the large intestine to get rid of it. So, speaking of the large intestine, that's what's next. There's a passageway from the small intestine. Oops, making my laser point. There's a passageway from the small intestine to the large intestine, and that's called the ileocecal valve. The ileocecal valve. It's called ileocecal because the last portion of the small intestine is called the ileum. Here, I'll zoom in. Ileum. And then the first portion of the large intestine is called the cecum. So there's the ileocecal valve, which is kind of that entryway from here to there. The large intestine, its whole thing is being a good water absorber. Absorb water. It's good for storage of indigestible stuff. Indigestible means that the small intestine or the stomach or any other process couldn't really get to whatever's in there yet. And then lastly, it's for the elimination of feces. The large intestine has 5 major regions. It's the cecum, that's this part here, the cecum. There's the appendix. There's the colon, which has some major regions. And then there's the rectum, and then lastly, the anus. The first portion of the large intestine is the cecum. And hanging off of that is the appendix. We talked about the appendix in the lymphatic system. And then once we get into the colon, there's the ascending colon that goes up, the transverse colon that goes across, the descending colon that goes down, and the sigmoid colon that kind of goes down, up, and around. Sigmoid refers to it's kind of like S-shaped-ish. Note here that what we're seeing is we still have mesenteries. And when we're looking at the mesenteries, the mesocolon is a special name for mesentery of the intestine. So that's the transverse mesocolon. That's the mesentery for it right there. And I'll show you some more pictures of mesenteries in a future video. This gray string-like thing that's kind of going across, that's a special new word. It's called tinea coli. Tinea coli means ribbon-like. It's a little bit of smooth muscle, um, which is like the large intestine's version of the muscularis externa's like longitudinal muscle layer, just to kind of like move everything. So it helps move everything down the line. The bumpies here, all these little bumps, are called haustra. One of them is a haustrum, 2 of them are a haustra. So plural is haustra. So 1 and 2, we call both of those haustra. And those are like those little sac things. The histology of this part of the GI tract is a little bit less complicated than the small intestine. There's still lots of absorption going on, or there's not a lot of absorption going on. So there's not a lot of circular folds, there's no villi, no brush border, but there is a thick mucosa with big old crypts in it. And those crypts contain lots of mucus secreting goblet cells. There's lots of mucus here. And that mucus is really important to help with the passage of feces and also to protect the walls of the large intestine from the bacteria who live there. So there's bacteria that live in the large intestine that can kind of set up shop in there. And the products that they secrete, we want to protect the wall of the large intestine from all that. So that brings me to my point. Why do we even have bacteria setting up shop in the large intestine? And those bacteria, they're called the bacterial microbiota. The bacterial microbiota or the gut flora. The large intestine has a bunch of different bacteria, and they get there by means of the small intestine, or those bacteria enter from the anus. But what do we get out of this relationship of like having bacteria in our gut? Well, and we actually get a lot of good stuff. I think of the bacterial flora in the large intestine as kind of like metabolism boosters. They're super helpful in performing two major processes: fermentation and vitamin synthesis. The process of fermentation is what some bacteria do to our indigestible carbs. So, to our indigestible carbs, um, these bacteria can ferment them and break them down into fatty acids and break them down into fatty acids, which can be used for energy in the body, right? But the thing is that fermentation also creates gases, which are released as flatulence. And flatulence can sometimes be stinky because of the composition of those gases. Flatulence increases as the amount of indigestible carbs that we eat also increases, like beans, for example. It's also important to have gut microbiota for the synthesis of vitamins. This is things like B vitamins, vitamin K, kind of thing, which are helpful for the body's functions. The second thing I think about when I think about the gut flora are that we want to keep those bad pathogens, the bad bacterias at bay. The thing is about the gut microbiota is that the good bacteria are thriving, and that helps suppress the bad bacteria because it's just a numbers game. There's just more of them. But also, our immune system we don't really want the gut bacteria in our actual body. The GI tract, right, is open to the outside, and we don't want to get the bacteria inside. And dendritic cells, they play a role in this and they help us out because they can trigger antibody mediated responses if those bacteria kind of like get a little too close to entering our systemic situations. There is a factor of the gut microbiota um, helping to balance out our immune system, like how many T cells we have of various varieties. But it's really important to know that we don't know a lot about the gut microbiota yet. We do know that there's more and more data coming out about how gut bacteria plays a really important role in weight, in disease states, and even our mood. And this is a really, really fascinating avenue for continuing and future research. So, gut microbiota, so cool. Okay, back to the large intestine. How does stuff move through here? So what happens is these haustra, they actually contract, and it's a segmenting thing. And throughout this process, we're absorbing water. So in the large intestine, we're absorbing water. But there's also stuff called mass movements. Mass movements, or like think of it as like mass peristalsis, it's a lot of peristalsis. It helps move the contents of the colon a few times a day, usually shortly after eating. And that's because there's food that hits the stomach that activates reflexes in the colon to move things along. And the descending colon is the one that goes down. So the descending colon and the sigmoid colon, um, they play a big role in storage of feces until those mass movements help push things down and out. And that process is called defecation when things get pushed down and out. So speaking of defecation, this is the process. It's three major steps. The first step is that feces move into the sigmoid colon, and then from the sigmoid colon into the rectum. And once that rectum, those walls kind of stretch, that initiates, so that stimulates stretch receptors in the rectum. And that activates and sets on fire like these neurons, right? And that activates a spinal reflex. It's going to activate the PSNS. When it activates the PSNS, we're going to do like the rest and digest thing, right? Um, so what this reflex does is it activates the contraction of the sigmoid colon. So sigmoid colon contracts, contraction of the rectum and of the sigmoid colon. It also promotes relaxation of the internal anal sphincter. So the sphincter, again, are muscles, right? So there's an internal anal sphincter and there's an external one. And the thing to note here is that there's different tissue types. Internal anal sphincter is smooth muscle, which means that this guy is involuntary. And then the external anal sphincter is skeletal muscle, which is voluntary. Which means that we don't always defecate right when that reflex activates. We can wait. So then once that internal anal sphincter is a little bit relaxed, that's this internal one, if it's convenient to defecate, according to your textbook, then we initiate a process in our brain. It's from the cerebral cortex. Remember, the cerebral cortex is for conscious mind, which means that's the voluntary part of our brain. The voluntary part of our brain, and then that voluntary part of the brain activates the external anal sphincter to allow then the relaxation and passage of feces. So it's a double layer. Once this rectum gets stretched, that ignites a reflex to then relax the internal anal sphincter to allow for that passage, but it's not going to pass yet because we still have to relax that external anal sphincter. And that's when the conscious mind comes in. The last picture in this slide set is just zooming in more on the last part of the large intestine. You can see some of those a little bit closer of the external anal sphincter, and then the internal one.